Hi, my name is Penny, and I'm working on a water quality project. Specifically, I'm focusing on fecal contamination. Coastal areas like Mobile Bay and Dauphin Island can experience elevated levels of fecal contamination because of human populations, unsupported infrastructure, and terrestrial watershed drainage. These inputs can transport pathogens that can have a negative impact on human health as a result of contaminated fisheries and recreational and swimming areas. In order to manage fecal contamination, the source of the feces needs to be known. That is, which organisms are contributors. As you can see from this figure, there are many potential fecal contributors to a coastal area. If the source of the contamination is unknown, the problem cannot be properly managed. Currently, the most widely used water quality methods only have the capacity to identify the presence of feces, but offer no information on who it is coming from or how much is really there. Therefore, advanced methods are needed to protect human and ecosystem health. Our project uses DNA-based methods, also known as molecular methods, to help determine the major fecal contributors to the Mobile Bay watershed. The first method we are using is called quantitative polymerase chain reaction, or qPCR. This method targets unique DNA sequences from particular organisms and has the ability to quantify the amount of DNA that is present in the sample. In this figure, you can see that each of these organisms has a different color DNA. This represents a unique DNA sequence, and this unique DNA sequence is what allows us to distinguish how much fecal contamination is coming from a particular source that we expect may be a problem in our system. In Mobile Bay, we are targeting human, cow, dog, pig, and bird through qPCR as our potential major contributors. Our second method is called environmental DNA, or eDNA. eDNA is all of the DNA within an environmental sample, such as water. This figure shows some examples of what eDNA encompasses, including free DNA, tissue, bacteria, feces. Anything that an organism sheds into the environment is considered eDNA. eDNA allows us to identify all of the potential fecal contributors to an area and minimizes the chance of missing potential contributors that targeted analyses like qPCR may not detect. The combination of these molecular methods as fecal source tracking tools can allow for the implementation of better management practices and protection for coastal communities, ultimately improving public health.